In this video, we're going to have a look at the role of T lymphocytes in the specific immune response known as cell-mediated immunity. Unlike phagocytes, which respond to all potential invading pathogens in the same way, lymphocytes have on their surface specifically shaped receptors which will bind to complementary antigens. An antigen is a marker protein on the surface of a cell which identifies it as being from a particular host. We could divide these into what are known as self-antigens, antigens which your cells have, and non-self, cells from any other living organism, including other people. The T lymphocytes are able to bind to a specific shaped antigen in the same way the lock and key hypothesis for enzymes works. Once this happens, the cell has become activated. This first stage of the cell-mediated response is known as clonal selection. This is because there are a number of different types of lymphocytes, each with specifically shaped receptors, and only one type of receptor will bind to one type of antigen. In the case of T lymphocytes, these cells have been produced in the bone marrow and matured in the thymus. They are only able to respond to antigens which are presented on the surface of a cell, not free antigens floating around in the body. So for example, a virus-infected cell will present viral antigens onto its surface. Cancer cells will change their antigens slightly as a result of mutations, so even though they are your cells, they do not have the self-antigens, and therefore your T cells are able to recognize them as being strange and odd. And the transplanted cells from another person will have different self-antigens to yours, so they are non-self. Once a T lymphocyte has been activated by binding to its complementary antigen, it goes through a process called clonal proliferation or expansion, where that cell will divide by mitosis to produce genetically identical daughter cells with the same receptor, and this just gives you a bigger fighting force against the potential pathogen. The next step is that the T cells which have been activated start to become differentiated. That means that they start to specialize. And there are four types of T cells: T helper cells, T memory cells, T suppressor cells, and T killer cells. The most important of these will be the helper cells, which are involved in activating a humoral response involving B lymphocytes, memory cells, which will give you long-term protection against the disease, and the killer cells, which are actually going to go and fight off any infection. T killer cells work by binding using their complementary receptor to the antigens on those antigen-presenting cells. So this could be a tumour cell, or it could be a virally infected cell, or it could be a non-self cell. It could be a transplanted cell, say from a kidney transplant. But these are non-self, so they're different to your markers. And when that happens, the T killer cell will start to release a series of chemicals, including perforin, which is a protein that sits in the membrane of this cell, digestive enzymes, or hydrogen peroxide, and basically just blows this cell apart, destroying it and hopefully any infection that it carries.